Hey everyone, I'm just gonna wait like 2-3 minutes for some people to come. Yeah, I might just get started because I feel like people are just watching the lab afterwards anyway. That's okay. So if we only have a couple of people, just feel free to unmute yourself anytime and interrupt me if you have a question. We can make it a very nice environment to ask questions in. No worries, just speak up. Okay, I'm gonna share my screen. Let's see if that works. Okay. All right. So today we're going to talk about single-cell RNA-seq, and tomorrow there will be a homework coming out. I think it's due April 11th. Don't quote me on that. It might change a little bit, but I think it's the 11th. The topic is single-cell RNA-seq analysis with Seurat, and there will also be a little bit about single-cell tag-seq analysis. So part of that is going to be covered by Philip next week, especially Maestro, which is the workflow developed in the LEO lab. And I'll be talking about Seurat today. And I hope it will be pretty hands-on. So first I'll briefly recap some of the concepts that were covered today about single-cell RNA-seq, and then maybe give you a preview of the lecture on Thursday. And then we'll go through a hands-on Seurat tutorial. And hopefully you can pull up the R Markdown file and you can read along, because I think that's the best way to learn. But Seurat in general is very, very well documented. So it's actually very nice to go on their site and look at all the vignettes they have. They're super well explained if you have questions about anything. So that should not be a problem. And then at the end, I know the homework hasn't come out yet, but I'll give at least a couple pointers, a couple useful functions that may be helpful when completing it. All right. So why are we doing single-cell RNA-seq? This is the canonical photo that people use. And I know we've spent most of the course so far on bulk genomics. And the reason why these fruit pictures are here is you can imagine if you sequence a tumor sample, you get a broad expression average of all the cells in that sample. And the analogy would be to a fruit smoothie. But a cell in, for instance, one area of the tumor may be quite different from a cell in a different area of the tumor. And that's where single cell genomics becomes useful. You capture signal at the level of each individual cell. So the analogy is that now you have all the individual pieces of fruit in that smoothie. And we won't get into the last one, but this is a really exciting recent innovation in the field, spatial genomics, where you can capture not just the expression matrix of single cells, but also their location. So that gives you a whole lot of opportunity to look at signaling between different cells, for instance. And this has been made possible by some new technology. I think it's called SlideSeq, if you want to read about that. So let's move on. Obviously, the body has many cell types. So there's a whole lot to explore when we're looking at single cell data. Here is the reason, in a little bit of contrived example, why it's important to not just have this broad average, because you could actually capture the wrong trend in your data by just using bulk RNA-seq. So in the left plot, for instance, you might deduce that gene A expression tends to decrease with higher gene B expression. But if you then actually stratify by cell type, you see that the opposite is true. You see that it actually increases. And this is only possible if you have that granularity of the single cell. So here I want to briefly backtrack and think about what we're doing, because it's maybe an intuitive idea to some of you, but for me, it was quite important to think about. So the caption is cells express the genes they need at the right quantities. So from the transcriptome, you get an idea of which proteins the cell is making. That gives you an idea of what the cell does and even what the cell is. So again, this may be obvious to you by now, but I want you to appreciate this idea, because perhaps when you learn about different cells in high school, you learn that a neuron looks like this, adipose cell looks like this, which looks different from a red blood cell, right? But there's this idea that the expression of the cell, the gene expression, actually defines what the cell is. And I want you to appreciate that. And let's summarize here. The gene expression profile is the calling card of a cell. So again, this may be obvious, but it's useful to appreciate, and it wasn't so intuitive for me. And once you have this idea, then you can imagine that with single cell RNA-seq, you essentially have a 20,000 dimensional address. So we can define any cell as a point in a 20,000 dimensional gene expression space. And in that space, you might imagine all the neurons close together, all the muscle cells close together, all the red blood cells close together. And this is what initiatives like the Human Cell Atlas do, which aims to give us a reference map of all cell types in the body. And this is going to be incredibly useful. If you have, say, diseased tissue sample, right? And then you want to compare that to healthy sample, you can now just tap into that big database of healthy cells, and there will be maybe tens of thousands of them perhaps for each cell type. I think the aim is to have 1 billion cells by the end of the decade or something like that. So it's very, very ambitious. So this is a preview in 2D. Since we can't visualize 20,000 dimensions really, there are all sorts of ways to compress that space down to 2D or 3D. Here I've shown all sorts of stromal cells, and we want to be able to separate these into different clusters so that we know which cell is which. So if you look at the granularity here, you'll also appreciate the usefulness of single cell RNA-seq. So in my own research, for instance, I'm looking at immune cell trafficking throughout the body. And the background knowledge there is that immune cells move through the blood, and then at some point they extravasate, or they go from the blood into a lymph vessel and eventually into tissue. And that happens in the post-capillary venules, which you can see on the right. They're annotated, and they have their own cluster. So now we have this subset of post-capillary venule cells, which would be very hard to isolate experimentally, right? But now we can just select them from the data set, and then I can look at interactions with other cell types, for instance. So this will be extremely useful. Okay, so I hope that I have convinced you that the skill is worth learning. Now let's quickly review how we get to a single cell data set. And I know this has and will be covered in lecture, so I'll make it somewhat quick. But the goal here is, again, to isolate individual cells. And it's not an easy task if you know how small a cell is. So take a moment to appreciate how amazing this technology is, where you capture a single cell in just a tiny droplet. And here I show you the 10x Genomics platform, which is the biggest commercial player in the space. And the details don't matter so much, but the important takeaway here is that we have a barcode added to all the transcripts from a single cell. And the goal of the barcode is to later identify which cell the transcript is coming from, right? So hence, you know, from which individual cell each transcript comes. And then there's the UMI, or the unique molecular identifier, which is important for normalization later on, because you can see in step 7, we are reverse transcribing the complementary DNA strand, which we then amplify using PCR. And we know that there is a PCR bias, which will amplify certain molecules more than others. I think it's called GC bias. So that's why you want that UMI, so that you can later normalize that. Okay, and feel free to speak up if I'm going too fast or something, but just gonna keep cruising through. Okay, so I showed you droplet sequencing, and that's pretty common, but don't think that's the only method. It's actually quite an expensive method, 10x Genomics. So people are looking at alternatives, and the most widely used, at least in labs, is something called split-seq, and it's a combinatorial method. So if just let's take a step back. If you want to identify thousands of cells, how do you quickly get to a large number like that? So the idea is that you do that through the power of combinatorics. If I were to combinatorics is just if I were to ask how many ways we could pair the people in this lab. I know we don't have too many right now, but if we had some more people in the lab today, then the number would get very large if you were to pair people in groups of 4. So that idea is at play here. So you distribute all the cells across different wells, and then you add a little bit, just a tiny chunk of barcode to the RNA transcript. You clear them out of the well, mix them all up, and repeat the entire process. Put them in the well again, add a tiny bit of barcode again, and after a certain amount of repetitions, each cell will have a sequence of tiny chunks of barcodes, and that will be unique probabilistically, and that will allow you to identify it. So if you want to learn more about that, I think this video is super good for the intuition behind it. It's quite fun. They explain it with ping pong balls and different cups. Okay, so here we see recap what I just said. I talked about the process of drop-seq and then the importance of the UMI to distinguish multiple copies of the transcript versus PCR artifacts. Now let's talk about the analysis, which is what we're here mostly to learn about. So there's a lot of steps in the analysis workflow. So we have this raw single-cell RNA-seq data that we just described how we got it. We have to generate a count matrix, run all our quality control, do normalization, do variance stabilization. We're not going to talk about that, but feature selection we will talk about. You might have batch effects, which you need to correct for. And if you have multiple sources of data, you might need to integrate those into one. There are also ways to do that. And we need to reduce the dimensionality. And then it's sort of up to you what your goal is. Yeah, so you can do the step that fits your need, or clustering or trajectory inference, which they do in developmental biology. Then you somehow need to visualize all that. And then, as if it weren't enough, the most tedious step is to then actually annotate which cell types you're dealing with on your cluster plot. So let's go one by one. So the first step in the preprocessing involves the generation of a gene expression counts matrix from the raw sequencing reads. So again, we have our complementary DNA sequence, a cell barcode that identifies the cell from which the cDNA was amplified, and then we have our UMI that identifies the RNA molecule. And the matrix that you then create comprises the gene expression values for the complete data set, organized by genes as rows, and then the cells as columns. Okay, and then quality control, right? So a lot of things can go wrong as you go through this delicate process of trying to capture these single cells in the droplet and then put it through the entire process, right? You can have cells dying, you can have multiple cells ending up in the same droplet, and that's not good. So you want to be able to control for that. One way that I'm showing here is mitochondrial counts. I think it's a sign of the fact that a cell is dying if it has a high mitochondrial fraction. So typically you would set some cutoff and exclude all cells above that cutoff. You actually have to be careful with that because you can't just blindly do what they do in the Seurat tutorial. You might have to think what your cell type is. Some cells, they simply have a lot more mitochondria. Some cancers actually use a lot of energy, right? And they might have a lot of mitochondrial dysfunction. So that's something to think about. You can't just blindly set that cutoff. And besides the mitochondria count, we also look at low read count if somehow you didn't capture a cell quite fully, or too many genes, right? If you capture 2 or 3 cells in the same droplet. Okay, then we need to normalize. So here we see in this plot is a good illustration where cell A clearly has a lot more reads. And normalizing this adjusts the data for the cell-specific differences in the total UMI count, and it reveals the true gene expression differences between the cells. So in this example, we have discrepancies in the library size, that is the total number of reads in a cell, and this masks the variation in the expression of gene 2 between cells A and B. on the left plot, it seems like gene 2 is about equally expressed, but then when you actually normalize it, it seems like it's has much higher expression in cell B. Okay, we've talked about batch effect in a different context, and here, similarly, it is a huge issue. So the left is no bueno, the right is still no bueno, but it's a little bit better. So yeah, both are not good. We want our cells to cluster by which cell type they are, not by which day of the week they got sequenced, right? So how do you know when you have a batch? You have to ask yourself several questions. It's likely that you do, unless you're very careful about your experimental design. But were all RNA isolations performed on the same day, for instance? Were all library prep done? Was that done on the same day? Did the same person do the isolation and library prep? Did you use the same reagents? Did you perform the isolation or prep in the same location? All these questions, if any of the answers is no, then you likely have batches. So to keep this in mind, if you at some point design your own experiment, try to design it in a way to avoid batches. On the right, they do that with only 2 replicates. You would likely need many more than that, because even in the right plot, you see that the clustering actually happens by color, which is the batch in this case, and not actually by the shape of the triangle or circle or the squares. All right. So yeah, if you're unable to avoid batches, they might be inevitable sometimes, then don't confound your experiment by batch. So what you should do is, again, split the replicates of the different sample groups across batches, and the more the better. And what you also can do is include this batch information in your experimental metadata. So then you can regress that out later during the analysis. Okay. Then dimensionality reduction. With such high dimensional data, we suffer from the curse of dimensionality. So this means that the metrics that one uses normally to measure similarity in gene expression patterns between cells, that kind of breaks down or becomes much less reliable when the space is very, very high dimensional. So you need to reduce the dimensionality of the problem. And you can reduce it into what it's called an embedding. And I know Josh Starmer has talked about PCA very well. That's a linear way to do that. Finds the linear combinations of genes that best capture the variance in the data. And there's a version of PCA. I think it's designed for single-cell RNA-seq data. It's called zero inflated factor analysis. We don't really have to worry about that for the homework or anything, but be aware that it's out there. And there's some debate going on here because, as you might expect, single-cell RNA-seq data has a lot of zeros. It's mostly zeros, any single-cell RNA-seq expression matrix. And this is partly due to the fact that some genes just aren't expressed in certain cells, right? And it's also partly that we in our reaction, we just don't capture that high of a percentage of all those transcripts. It might only be 10%, but it's like kind of a high, wide confidence interval, but it might be between 10 and 50% of those transcripts that you capture. So you're just missing a lot there. And so you don't necessarily know whether a zero is due to the fact that the gene is simply not expressed, or whether it's due to missingness, right? So there have been people thinking about how to then impute those zero values. So this is something you should be careful with. You have to know what you're doing, because when you're imputing data, you're essentially changing the data. You're modeling your missing data. And I'm also pretty sure we don't need to do this for the homework, but it can improve your downstream analysis if you do it right. There are now lots of deep learning approaches for this. There is a widely used tool called Magic that uses information from cells that are similar to then impute missing values. But yeah, maybe don't worry about it right now, but be aware that this is something people do. Okay, then we have to cluster. So here we're grouping based on similarity in gene expression. And finding the optimal number of such clusters can be challenging. So there are several methods to do it. Some methods are able to preserve the local structure. So that is the within cluster distances between cells of the same type. Others will preserve the global structure, which is the between cluster distances. So there you have then you can compare if a cluster A is farther away from cluster B than cluster C, then cluster A is essentially more similar, or sorry, cluster C is more similar to cluster B. So, sorry, I hope that was not too confusing, but then you can compare cluster distances essentially, and otherwise you can look at the dots within the cluster. If they are far away, they are a bit more dissimilar. If they're close by, they are more similar. Okay, this was the last step, and this ties into my own research a little bit. It's just cell type annotation. It can be quite time consuming, although there are now semi-automated and automated ways to do it. But the idea is that you have all your different clusters, and then you have to find genes that are uniquely expressed in each cluster. Match those genes to usually a list of canonical cell type markers. So you might know that a certain gene, let's say CD8, is key in CD8 T cells, right? So if you know that, you can see which clusters highly express CD8, and then you just, if they also match a bunch of other canonical cell type markers, then you call them CD8 T cells. You can accelerate this using functional pathways and gene ontology. And I am actually doing a project myself on doing automatic annotation, which I find quite exciting. We don't have to worry about that. Okay, so all these steps above that I just described can be done within a single workflow, and that's what we're going to talk about. Seurat is the most widely used one, and it's fully in R, very well documented. Then there is ScanPy, which is the Python alternative. Maestro is developed in the Leo lab, and Philip will talk about that next week. There's also a cloud-based alternative now that's developed at Broad Institute. I think it's called Cumulus. That's very new, but it might actually take off and replace one of these. So let's all try and open up our RMD files, and let's go have a look at Seurat. I'm basically walking you through the basic clustering tutorial that's also on the Seurat website. So yeah, make sure you have it open up. I'll give you a minute to do that. And while we do that, I think Georges Seurat, if I'm pronouncing that right, he was a expressionist artist. And my hypothesis is, I haven't checked this, but is that all the tiny little dots he used in his paintings resemble the dots that you might see in single cell RNA-seq plots. But there may be some entirely other reason, but that's my thought process. Okay, hopefully you have it all open up. So Seurat updates continuously. So please stick with the Seurat website's explanations and documentation, because on Stack Overflow, people might give you a solution that's using Seurat 2.0, which is actually quite different from 3.0 and 4.0. Okay, so we'll be analyzing a data set of PBMCs, or peripheral blood mononuclear cells. This is available for free from 10x Genomics. There are 2,700 cells sequenced on the Illumina NextSeq. And let's start by reading in that data. So there's a function for that called read 10x. What we'll then create is a Seurat object. This object is very central to the Seurat workflow. It serves as the container that contains both the data, like the count matrix, and also contains analysis like PCA or clustering results. So it takes something to get used. It's not just a matrix, not just a count matrix. You can access the count matrix, but then you can also access PCA results, clustering results. So that's very, very useful. It all happens within this object. All right, let's move on. So it tries to store these matrices in a sparse manner. You can see that by all the dots instead of zeros. So those dots just represent zeros. Since most values, as I said, in the single cell RNA-seq matrix are 0, this is very useful because it significantly reduces the memory of this matrix, and this leads to much faster computations. One thing to know, this may not be relevant for the homework, but if you go and use Seurat later on, if you try to use some packages written in R, those can, under the hood, convert a matrix into a dense convert a sparse matrix into a dense matrix by forcing it by saying by calling as dot matrix or something. You have to be careful because that can quickly blow up. A dense matrix may be 30 times as big in size, and the sparse matrices are already big. They can already be gigabytes. So then it might, you might have the forever spinning wheel, and it may take forever. So you have to be a little careful when using outside functions and saving matrices. Okay, now QC, simple as that, but the one line of code at the bottom, at least for the mitochondrial genes that I talked about. So again, we want to filter out low quality cells or empty droplets, which have very few genes, and then cell doublets may have an aberrantly high gene count. And then we also want to take care of the total number of molecules detected within a cell. That correlates strongly with unique genes. Okay. Here below, we are looking at the percentage of reads that map to the mitochondrial genome. So low quality or dying cells often exhibit extensive mitochondrial contamination. And we can use the function called percentage feature set, essentially pattern matching the MT string, which is just in front of a regular gene. If it has a prefix MT, it means it's a mitochondrial gene. And yeah, the percentage feature set just calculates the percentage of counts originating from mitochondria. So then you can conveniently access this by doing add metadata. The percentage feature, what's it called, percentage feature set function will just add it to the metadata. And this metadata is very useful if you want to look at all, just the metadata of your matrix. You can just conveniently keep track of all of that. Okay, so now let's think about, during this quality control, what kinds of cutoffs should we set? Again, we don't want too many or too few RNA counts. We don't want too high percentage of mitochondrial genes. So how do we think about that? And the answer is not so straightforward. There are some rules of thumb, but one way to start that process is by doing a violin plot. And here you can see that most cells fall within a certain range. And if there are some aberrant values, there likely something went wrong. Like in the left plot, if there is a y-axis value of over 3000, that's probably something went wrong there. So we're set some cutoff that will get rid of those observations. And let's see where we do that. Oh, first, here's another way to plot essentially the same concept. You just plot them against each other using feature scatter. And here we actually do the filtering. So once you come up with a cutoff that you think is right, you can use the function called subset, which literally helps you take a subset of the data. You can think about it like if you have ever used pandas in Python, it's just filter. Okay, now we're to the normalizing step. After we've removed all these unwanted cells, we now need to normalize. There's a default method in Seurat, which log normalizes the feature expression measurements for each cell by the total expression. Then it multiplies it by a scale factor, which you can also set as 10,000 by default. And then it, as I said, log transforms the results. The top line here is just showing the default values. You don't have to include them, but only if you want to change them, you should edit them. All right. Then what they've found, how do I say this? They found that focusing on a certain subset of genes in downstream analysis helps to highlight signal from the noise. So there might be a lot of genes that just don't really vary that much across cells. Maybe all cells use them or all cells don't really use them. It's really where the action is happening is with genes that have a high variance between all these different cells. That's where you can really glean some insights. That's the idea. So just taking a subset of those genes to your downstream analysis can be very useful, and it's also quite a little bit faster. So by default, you can do a variable feature plot and then use the function find variable features to, by default, return 2000 features per data set. Features is just a gene. And then, again, we take these to our downstream analysis. Okay, if you then want to do, especially PCA, you have to scale the data. It's also necessary for a lot of machine learning methods. Scaling just means making the mean 0, making the variance across cells 1. And this is important because it gives equal weight so that highly expressed genes do not dominate the later downstream results. So there's a simple function called scale data to do just that. Okay, this is how you run PCA in Seurat. If I were Josh Starmer, I would say something like poop, 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 if you've seen his lecture. So I will leave that to him. I'll leave you to watch his PCA video. He's much better at explaining it than me, but Seurat makes it quite convenient. So once you've actually ran PCA, you can look at the results in a few different ways. One is to simply print the genes with the largest coefficient within each principal component. So yeah, what does this do? These are just the top 5 genes with the most positive and then the most negative coefficient in each of the significant principal components. So that's one way to see which genes matter, which genes are having an effect here. And note that each PC is just, when I say coefficient, remember that each principal component is just a linear combination of all the genes, just a weighted sum essentially. It's like some scalar times CST3 plus some scalar times thyrobp, et cetera, et cetera. So if the coefficient value is very high, this gene has a lot of effect. Okay, that's one way. On the left plot here, you're essentially just plotting that, but we just printed. And on the right, we have a, as of yet, uninformative dimensionality reduced plot, so it's not so informative just yet. But let's move on. This is something you will need to use in the homework. It's called Jackstraw. So this can take a long time at first, so be aware of that. It's probably nothing wrong. So you have to do Jackstraw, then score Jackstraw, and then you can do a Jackstraw plot. So why is this important? It provides a visualization tool for comparing the distribution of p-values for each principal component with a uniform distribution, which is the dashed line. So maybe the details don't matter so much, but if, they describe it as a resampling test, which is a Jackstraw procedure, where you randomly permute a subset of the data. You then rerun PCA on that subset and construct a null distribution of feature scores, and then you repeat this procedure. So then you can say a significant principal component is one that has a strong enrichment of low p-value features. So if I'm not explaining it very clearly, please just look at the Seurat documentation. I think it's quite clear and you can read up on it a little more. There's a whole paper on the Jackstraw procedure, but know that these are the functions that will be useful in the homework if you're asked which principal components are significant. This is the more common way to look at which number of PCs you should use. So it's hard to, in a quantitative way, say, this is the number of principal components I should use. This elbow plot is useful because we want our principal components to capture a lot of the variance, or on the y-axis here, the standard deviation. And usually one might say the point where it elbows this plot, where essentially you get very little value from adding an extra principal component, that's where we set the cutoff. Again, it's a little ambiguous whether that's 6 species here or whether that's 10. Usually it doesn't really matter so much, actually. Just if you know that if you have a very large data set, then 10 might not be enough. I've heard the rule of thumb of 30 principal components for larger data sets. So just remember, this is only a data set of 2,700 cells. All right. So clustering, also very conveniently implemented using the find neighbors function. So this embeds the cells in a graph structure. For instance, k nearest neighbors with edges drawn between cells with similar feature expression patterns. And then it attempts to partition this graph into different communities or clusters. So for the find clusters function, you should take note of the resolution parameter, because this is what will influence the number of clusters you obtain and the number of cells that are assigned to each cluster. And I think in the homework, you'll be asked to compare your plots based on different values of the resolution. You'll see what I mean. But here I show you an example. So on the left, you see a resolution of 0.4. On the right, you see 0.8. You see on the right, you see a lot more clusters. And the intuitive idea is that this granularity, you can make it as granular as you want. Do you just want to annotate a cluster of T cells, or do you actually want to annotate CD4 plus T cells, CD8 plus T cells, right? That is up to you. And by tweaking the parameters, you can tweak the granularity of your clustering. So besides PCA, there are nonlinear dimension reduction methods, of which UMAP is the most used one. TSNE is another example, although it's not used as much anymore. So the goal of these algorithms is to learn the underlying manifold of the data in order to then place similar cells together in a low dimensional space. So the idea is this high dimensional data exists on a lower dimensional manifold, which is still useful. So cells within the graph-based clusters should co-localize on these dimension reduction plots. And as input to UMAP or TSNE, I would suggest using the same principal components that you used for the clustering analysis before. There's also a very useful blog post that I linked to here. You can click on in the PDF, which explains UMAP very well, and the difference between UMAP and TSNE, and why TSNE has kind of gone out of fashion. Because in short, the issue is the between cluster distance is not very interpretable. So as it says there, it's hard to say if clusters far away are less similar than clusters close by. And that's an important trait that you would want to be able to interpret. Okay, so once this is all done, Seurat can help you find the markers that define these clusters via differential expression, which we've seen before too. So by default, it identifies positive and negative markers of a single cluster and compares them to all other cells. And the function find all markers automates this process for all the clusters. But you can also test groups of clusters versus each other or against all other cells. Then the minimum min PCT argument here requires a feature to be detected in a minimum percentage in either of the two groups of the cells. And then you can set a fold change threshold as well. If you set these to 0, this process will take a really long time. So you do want to set it at some value that's positive. All right, so yeah, now we are able to do differential expression analysis on different clusters that we have, which we can visualize that in a way where we say, looking at all the 8 different clusters here, where is this canonical marker gene that I have? Where in which cluster is that expressed? So I'm not exactly sure what MS4A1 and CD79A are, what they do, but we know that they are expressed highly in cluster number 3. So that will tell you something about what is cluster number 3. Then there's a nice function called feature plot where you can just put in a vector of all these canonical genes you're interested in, and it will show you all the areas on the UMAP plot where they are differentially expressed. And then the process is to match these clusters to the canonical markers. Again, we won't have to do that in great detail, and it's quite time consuming. But the idea is that ultimately you end up with a beautiful 2-dimensional plot where you can separate out all the different clusters by cell type. And so, as I said, that's still a human endeavor as of today, but I am very excited about semi-automated and automated ways that can make that easier. All right. This is something I highly recommend if you want to take single cell sequencing a little more seriously and perhaps do your own analysis. It's a really, really useful, well explained, no background required intro to single-cell RNA-seq analysis offered by our friends at the Harvard Chan Bioinformatics Core. I've attended some of their workshops, and I think they are really, really, really useful. And it's all available on GitHub, so you can just work through the example code that they have. And they have basically all these above steps that I just walked through really, really quickly in a lot more detail. And yeah, I highly recommend it. This is a shameless plug. So the creators of Seurat are hosting a conference or a day, single-cell genomics day on Friday. You may think, Martin, I don't have time to go to some conference in the middle of the semester. And that's fair. But if you're doing research in single-cell genomics of any type, I'd actually recommend watching even just the first presentation. It will also later be on YouTube, don't worry, by Rahul Satya himself, so the developer of Seurat. He gives an overview of the progress in the field that's been made in the last year and where it's headed. And I watched the last couple of years videos, and he's almost always been right. While this field moves so fast, he can predict, oh, these papers that come out, these are going to be huge. These are going to be very important the next year. And then he checks back in the next year, and it turns out he's almost always right. So he has a good grip on what's the cutting edge, what are the important papers that are coming out. And if I personally, the only reason I found out about a tool I'm using called Cell Phone DB, it's not important, but it's for cell-to-cell interactions, was through this video. So if you're doing any research involved single cell, I recommend you at least watch the first Rahul Satya presentation. Okay, this, not sure I have to go through all of this because the homework isn't even released yet, but I'll walk through it very shortly. Part 1 will require you to use Maestro, and it's quite well annotated on the Leo Lab GitHub page, I think. So this is the canon command, the cluster command that you will have to use. And again, it's one of these exercises where it's just a mix and match of the right directory name and file name into the right spot. Same thing for the initialization of the single cell tag-seq. Same thing. Part 2, that's where what we talked about today comes in. You have to use the Seurat functions I talked about. Here I'm listing which ones are may or may not be useful for each problem. And I hope you find this useful. And same for part 3. And then number 4 is quite exciting. It's about integrating single cell RNA-seq and single cell tag-seq data. So yeah, here I'm also providing some hints for that. And with that, let's see if there are any questions. And if not, thanks all for joining. Let me stop my share. And let me stop the recording.